It gives me great pleasure on behalf of Macmillan Education to introduce Dr. Durant to you. We're so grateful for this. I've already listened to this presentation once today and it's fascinating and really informative and helpful. Dr. Durant is a biology expert who gained her doctorate from Imperial College of Science, Technology and Medicine in London. She is a former Caribbean Examinations Council Assistant Chief Examiner, Cape Biology. Dr. Durant has changed in educational measurement, assessment, evaluation, and has taught at both secondary and tertiary levels in the UK and the Caribbean. So without further ado, thank you so much, Dr. Durant.
So, good afternoon, and I'm so pleased to know that you are taking your time to be here to find out more about the Biology for CAPE examinations textbook, both as a physical textbook and available online, and to join me to take you through the textbook and also some difficult topics, two difficult topics in biology, and also how you can really use the resources that are available to make the most of your time, whether it is blended or if you have to study from home online. So, just an overview of the webinar. We will review key features of the textbook, coping with online learning for both teachers and students, strategies, methods, and support. We'll have a review of some challenging topics in cat biology, which are calculations, chi-square, important definitions, and some examples in real life. And then the immunology and vaccination, because as we know, these topics are all in the news and everybody is keen to find out what immunology is all about and vaccination. And then um, we will have a look at the video, and then a self-study worksheet will be available for you through Macmillan. So the key features of the textbook is available as a physical book and also as an electronic book that can be downloaded from Macmillan Caribbean eBooks. And in addition to purchasing the entire textbook, there is an option to allow you to rent the textbook for 3, 6, 12, or 24 months. We know that these are difficult economic times, but we still have to continue with our studying. So, by renting the textbook for a short period of time, that might be more economical for some. And that way, you would be able to still cover your work and be able to get all of the benefits of the wonderful information involved in the text. Now, the Biology for CAPE examinations is a recommended resource in the CAPE biology syllabus, the CXC syllabus, and it covers both Unit 1 and 2, the school based assessment in a single book. So, normally, what we call lower 6 and upper 6 are the 2-year course for your certification in biology. This book covers all of that in a single book. The book has learning objectives with a summary for each chapter, and we have lots of Caribbean examples with photographs. So we use references in photosynthesis and flowers and flower um, morphology and learning about the outdoors and ecology. We use examples like mangoes and the coral reef and sugar cane. All of those are examples that we are all familiar with. The book also has highlighted reminders of linkages between topics because we know that biology is an integrated subject. So even though you learn about mitochondria at the beginning, mitochondria are important when you're learning later on how the heart works. If we learn about water and water's properties, but those are important for the transmission of things in photosynthesis. So the book has important linkages highlighted to help you to understand that biology is an integrated subject. The book also offers questions for topic review and the answers are provided, sorry. And they are essay questions similar to what CXC would set in your paper too at the end of each chapter. And we all know that practicing questions is important for you to determine if you have understood a particular topic. There are study tips for exams and laboratory experiments with expected results. These experiments would have been designed with, working in the laboratory, in the classroom, or at your school in mind. And it includes expected results because sometimes you might be working on an experiment and you are not certain if or certain what the result should be. So the experiments give you an idea of what is involved. Also, we have some easy SBA experiments for our students to perform on their own, and the video at the end will give you an idea of what that looks like. The last chapter is dedicated to practical biology techniques for SBA, things like how to do a biology drawing, how to do the labels properly, how to do your analysis and interpretation. And there are also criteria that can be used by the teachers. And this is also great for students as well because you will have an opportunity to see how your teacher is going to be grading your school based assessment or grading any projects or any work that you might be putting in. So, managing work in online school for the teachers, we know that we have to offer active learning experiences to keep children engaged. And it is important to set clear expectations for work, but keep in mind that students may require more flexibility and understanding. We know that working from home is not easy for adults and it can be challenging for our students as well. So, we have to make sure that they are able to keep up with what needs to be done in order to make sure that they fulfill the requirements. Also, we do know that every student might not have access to stable internet connections and wide bandwidth, so we have to be able to work with them as well to make sure that they are coming along. It may be necessary to adjust the time allotted for the school-based assessments to ensure that they're completed properly, and structure assignments so that grading and feedback can be managed without becoming overwhelming in this online environment. And it is necessary to for students to have the opportunity to ask and answer questions and work in groups, even if these groups are virtual with their friends and colleagues from their classes, so that they can tackle problems using real data. Over the course of the last year, we would have been introduced to the concept of fake news and fake information and misinformation, and it is important to use credible online sources that could support data collection and analysis because we can use data from the scientific literature to actually do analysis and interpretation if you do not have the opportunity to do this yourselves. And credible online sources include places like universities and museums, World Health Organization, PAHO, Smithsonian Institute, all of those, Royal Society, all of those places have credible online data that can be used for experiments or for determining any information that is needed. Now, in planning the XBA or laboratory work in online school, if you share an online lab worksheet in advance with students, it helps them to record data easily and keep themselves organized. And students in an online laboratory class can be divided into groups so they can use their actual lab experience, because some students would have had the opportunity to do actual labs. And if they are in a group with their colleagues who might not have as much experience as them, they can help them as well. um, in the online world, you can give an overview of the lab, its objective, and give a few questions on the lab topic, or ask simple questions like, what is the purpose of a control in an experiment? And let the students just think, okay, why do I need to include a control? Or why does the method have one activity before another? Why are we doing it in a particular order? And um, students can answer these questions individually or in groups, get together so that they are still engaging in order to get a similar experience to the physical laboratory if they were at school. And lab safety procedures and recording information on your worksheet is important, because if things are kept organized, it is always good. So for data collection and analysis, the teachers can prepare photos or demonstration and share a data sheet of actual results, or create a video of a technique and annotate the key steps, and the students can just watch this and are determine how the experiment would go and collect their information in that way. So students can also analyze data and answer their questions using the worksheet. And we know analyzing data includes distinct skills that become easier the more you practice. So these are all important areas that we have to consider when we are trying to get students through in the online school. Learning how to review literature and critically read peer-reviewed journals is important. And in the online lab, all the students can take a simple journal paper, a paper from Nature or Science or any of those reputable journals, and read the abstract and the introduction, and use those to determine something about how an experiment is conducted. Simple reading and just looking at information and discussing it with your colleagues or with your teacher helps you to see how scientists conduct experiments and how they come upon their conclusions. And this can be done, as I said, in the class, or it could be an assignment, and students can do it themselves. We all I spoke before about extracting data sets from published literature, and with these experiments, students could perform an analysis and focus on data interpretation and analyze it just as if they had collected it themselves. And we know because of the pandemic, we have to use the outdoors more, and the outdoors is a great place for experimentation, observation, and data collection. For biology, we can look at trees, the girth of trees, we can look at leaves, we can look at dirt, we can look at insects underneath the dirt, we can look also for ecology and conservation. We can choose many things from the outside in order to um, collect information by observation and use it to collect data. It, we can also consider having students create self-narrated video submissions as part of their work. We know that students love social media, and videos are a big part of social media. And creating a self-narrated video submission is a way to get them interested and keen on some aspect of biology. It could be part of their project, or it could be part of some other activity that they have to do. And this will certainly, I am sure, be a hit with most students. Um, Planning an assigned lab time for students and having a channel open for students to come in and ask questions is important, because working at home alone is okay, but when you meet a challenge, it is important that you have someone there who can always assist you and give you an idea of how you can solve the problem, so you would not feel as if you are on your own. So for students, we have to treat your online class just as you would a physical class in school. Designate a little space at home wherever you normally sit. And if you have a space, gather up your notepad and pencils in that spot, so you know, okay, this chair or this desk or this little area is where I would normally have my school. It is easy to say, okay, I am watching the teacher on the online and I can just go back and look at it, look at the video. But in online school, taking notes, just as if you were taking notes in class, are important, and this makes revision easier. Ask your teacher questions in online class just as if you were in the regular class. We know that social media, if you have your phone and it pings all the time, you want to take a look, but resist the urge to go on social media while taking classes, because this is going to distract you from learning. And we still have the same limited time in order to cover the syllabus. So if you get behind by not paying sufficient attention, then you are not going to live up to your full potential. In between classes, get up and walk around to stretch your limbs and rest your eyes from the screen, because we know that that is something important to keep you engaged. So just walk around in between and make sure that you stay organized and manage your time so you can fit in all your assignments, not only those from biology. So for the students, you can download the CAPE Biology syllabus from the CXC website. It's completely free, and you can use it to track your progress through the topics. We know that the SBA is to help you to develop your knowledge and skills, such as analysis and interpretation, manipulation and measurement, observation, recording and reporting, and planning, designing, and drawing. From the CXC website, you can also download the CARE SBA Form 2021 and review the appendix, which shows how marks are allocated for the biology skills. And the end of the syllabus, which you download free, has specimen examination papers with mark schemes that give you the answers. So, what I suggest is when you have completed a topic, just have a look through the specimen paper and attempt a question on the same topic, and then check the answer that is given at the end of the syllabus, and it would give you an idea of if you are on the right track or if you need to go ahead and review the information once again with your teacher or with your friends to see if you can understand where you have gone wrong. So this is the appendix from the CXC SPF form, which I said can be downloaded from the CXC website. So when you are doing your experiments, and for example, planning and design is a skill that is being assessed. You know that for the hypothesis, you get 1 mark, for the aim, 1 mark, for writing out your materials and apparatus or putting in a little diagram of it, that's 1 mark. The variables stated, you see there control, manipulation, responding, you see a mark is allocated there. For your method, clearly outlining how manipulated variable will be changed and measured, and clearly outlining how the responding variable will be measured. So when you are doing your experiment, you know, okay, this is what is going to be looked at for determining my mark at the end. So I have to pay attention to make sure that when I'm doing my write up, I include these things for your expected results and what you have done, and then what precautions, limitations, and assumptions you included, and how you have stated them. And you see there at the end, you can see how the total of 12 marks was arrived at. For the analysis and interpretation, you see here also we have the discussion of how the marks are allocated here. So by downloading this item free, again, you have an idea of how your particular um, experiment will be judged, and then you can organize yourself to suit. Okay, so biology for CAPE examinations has a chapter dedicated to practical biology. So we have a chapter that gives you an example of, say, um, how to measure mitochondria, how you do the calculations to say what is the size. We also have in that how to do your biology drawings and put the labels. So the practical part of the biology is well explained and examples given in the practical biology chapter. Now, many students, might say where you want to work on your own and now you're at home, you're working alone. However, working alone can be challenging if you meet upon areas that are very difficult. So I suggest that you keep reaching out to your teacher using available channels, and also try to form a work group with other persons, other colleagues, so that they can help you work through. Otherwise, it could be isolating just working on your own. So let's just go through some challenging topics. We'll look at genetics and reproduction, and also immunology and vaccination. So we know the Punnett square is a table which is used a lot in genetics. It shows all the possible outcomes for a genetic cross between two individuals with known genotypes, and it is used to predict the genotypes of a breeding experiment. We know that Mendel did lots of experiments with pea plants, and we are going to just have a look at a little experiment with that. The Punnett square shows the proportion of different allele combinations in offspring and the phenotypes of offspring. And for all the ones studying biology, you would know that the law of independent assortment states that alleles of different genes assort independently of each other during the formation of gametes. And this applies to genes that are on separate chromosomes, but not necessarily genes that are on the same chromosome, because we know that chiasmata are formed sometimes. And the chi-square test is a statistical test that predicts the differences between groups, and it is used to determine whether these differences are significant. Now, we just go through a short experiment here. Two pea plants, just like how Mendel did his crosses. Two pea plants that are heterozygous for yellow seed color are crossed, and yellow seed color is dominant to green seed color. And we crossed two plants, and the 180 seeds that were collected, their color was observed. So the ratio of yellow seeds to green seeds expected by chance is 3 to 1. And we can see that here from the Punnett square, which has the male gametes and the female gametes. And you can see here that the green seed is when we have the two small y, which normally represents the y. Here, the yellow seed color y is dominant, and the small y is for the recessive here. So from the number observed, we collected 180 seeds, remember? So we collected 129 yellow seeds and 51 green seeds, which is a total of 180. And the number that would have been predicted using our 3-to-1 ratio would have been 135, which is 3 times 45, and 45, which is obviously 1 times 45, to give 180. So if we use the formula, find the chi-square, and we insert our numbers from the previous table, our chi-square value comes out to 1.0667, and this number is the chi-square value is affected by the degrees of freedom statistic. The degrees of freedom is equal to the number of groups minus 1. So we had two groups, green and yellow peas, and the degree of freedom is 2 minus 1, which is 1. So the threshold value of the chi-square that is applicable is 5%, and 5% is the traditional number that is used for biology experiments. And the chi-square value for 5% at 1 degree of freedom is 3.841. So since 1.0667 does not exceed 3.841, there is no difference from the expected 3-to-1 ratio. So we know that the seed color is segregating according to Mendelian inheritance. Now, we are dealing with peas here, so a 5% probability is okay. That's the normal thing. But if we were dealing with a pharmacology experiment where we were looking at a drug or we were looking at something dealing with health, then in those particular instances, instead of 5%, we reduce it to 1% to make it more stringent, to make sure that we do not come up with something saying that an activity is happening by chance when it is not. So, we are in the Caribbean, and I'm certain that most people are familiar with guavas. The red flesh color in guavas is dominant to white flesh color, and this is controlled by one gene. And dominance occurs when alleles produce the same phenotype, whether the pair of alleles is heterozygous or homozygous. And we saw that previously with the green and yellow peas. Incomplete dominance occurs when alleles are not fully dominant or recessive to one another, and heterozygotes show an intermediate phenotype. And we see that a lot in skin color. I'm sure you have friends of all skin colors, as do I. And codominance occurs when two alleles at a particular locus of the DNA produce two different phenotypes which can both appear in heterozygotes. And an example of this is the ABO blood group. And information on this is included in the textbook. So another area in genetics and reproduction relates to things like epistasis, and epistasis is an inheritance pattern in which one gene mask the expression of another gene, and the second gene is at a different locus, and each gene in epistasis is inherited independently. And this happens in fur color in mice, where mice can have gray fur, whitish colored fur, they can have banded fur, and this is an example of epistasis. And sex-linked genes in humans, red-green color blindness is an example of a sex-linked gene. And this little chart here that I took from the USAID, if you don't have red and green color blindness, you should be able to see the number 73 written there in that box. And sons normally inherit sex linked genes from their mother, because you know that gentlemen have X and Y, so they only have one X chromosome. And many gentlemen are suffer from or have the condition of red green color blindness. Hemophilia is another example of a sex linked gene. So let's now just move on to immunology and vaccination. We know that the body has ways of defending itself against pathogens and disease causing organisms. The skin, mucus, and cilia, which are in the lungs, work as physical barriers to prevent pathogens from entering the body. But sometimes you know that pathogens get past all of these, especially if you have a cut or you have some place on your body that is open, or just into your mouth or into your nostrils. And we have learned a lot about that since we had COVID. When a pathogen does infect the body, our body's defenses, which is the immune system, are triggered, and it attacks and destroys the pathogen. Now, each pathogen is made up of unique subparts, and the subpart of the pathogen that causes the formation of antibodies is called an antigen, and antibodies are produced in response to the pathogen's antigen. And each antibody is trained to recognize one specific antigen. So at any one time inside of your blood, there are millions of different antibodies which can recognize different antigens. And this is how the body helps to keep us safe. So when the human body is exposed to an antigen for the first time, it takes time for the immune system to respond and produce antibodies specific to that antigen. And we look at that here now. So the diagram on the left is taken from Biology for Cave Examinations, the textbook, where we can see if we expose ourselves, our bodies to an antigen at day zero or day one, there is a little lag before we get a primary immune response after the first exposure. But then, by the time you are exposed to the antigen the second time, there is a rapid increase in your body where the memory cells formed during the first exposure are activated by the antigen, and the secondary immune response is faster and higher than in the first primary immune response. And this is just like us. When you are learning to do something for the first time, it takes you a little while, and you might be apprehensive and you might not be getting it done quickly. But the second time you have to do something, you say, I know exactly what I have to do. So that means that I can move ahead quickly. And it is the same thing with your immune system. So many of us, as children, we would have been vaccinated from things like measles and chicken pox or the flu, and our bodies would have still antibodies circulating to protect us if our bodies ever see those particular microorganisms inside. But if we have a new pathogen, just as the COVID, there is no antibody, and that is where the challenge arises. And that's why we have the vaccinations to help us to be able to manage our bodies, to be able to protect us. So, vaccines contain weakened or inactive parts or antigens of a pathogen that triggers an immune response. Vaccines can also contain the blueprint for producing antigens rather than the antigen itself. So, vaccines will not cause a disease in the person receiving the vaccine, but it will prompt their immune system to respond as if it met the actual pathogen. And vaccinations involve receiving a vaccine via a needle or drops by mouth. And I remember as a child receiving the polio vaccine as drops on a little block of sugar. Immunization is the process of both receiving a vaccine and becoming immune to the disease as a result. And immunological memory is the basis for durable protective immunity after infections or vaccinations. So things like tetanus, when you get uh, the vaccination for tetanus that can last up to 10 years in your body. Some other vaccinations, such as the measles and the chicken pox or MMR vaccine, those also last sometimes for years. So, all that time inside of your body knows that if I meet this again, I am able to respond because it has memory cells there. The most important ingredient of the coronavirus vaccine is the messenger RNA to instruct your cells to make the coronavirus spike protein. Coronavirus uses its spike protein to attach to molecules on the outside of the cells to get inside and reproduce itself. So, messenger RNA from the coronavirus vaccine is taken out by your ribosomes and translated into many copies of the spike protein. And the messenger RNA is broken down inside of your cells, but the newly formed spike protein, which is the antigen, is released from the cell. And the cells in the bloodstream come in contact with this antigen. They know they haven't seen it before and they raise the alarm, and that's how your body gets ready to make sure that it can prevent the COVID virus from taking over your body and making you sick. Messenger RNA vaccines require two shots. Remember, we saw the table before where you have the primary immune response and the secondary immune response. And during that waiting period, memory cells and antibodies are generated. And the COVID 19 vaccine, like all vaccines, relies on immunological memory. And in response to the vaccination, the humans make coronavirus specific antibodies and coronavirus protein specific memory cells. So just let's have a look here at the how the messenger RNA vaccine from Pfizer and Moderna, what it looks like, and the COVID 19 vaccines for the RNA use lipid nanoparticles to take the molecules into the cells. And from the diagram that we can see here, we can see how it is composed. It has the phospholipids, just like how we have the phospholipid bilayer with the hydrophilic heads and the fatty acid hydrophobic tails arranged, and this is arranged in a circle. The lipid nanoparticle is arranged in a circle, just like if you had a micelle or a phospholipid bilayer wrap back on itself. So these The vaccine, when it is injected into you, it has the messenger RNA on the inside being protected by the phospholipid bilayer, and then that messenger RNA is taken up by your ribosomes, and then the coronavirus spike protein is created. So just let's have a look at how that happens. So we have um, the vaccine on the outside there in the purple, and that is injected, and the spike protein messenger RNA goes into the cell, into the cytoplasm, and then the ribosomes make an amino acid chain, which is the which then folds itself into the spike protein, and then these spike proteins then present themselves on the outside of the cell and then also into the bloodstream, and in that way, your body says, I have not seen this particular spike protein, which is an antigen. I have not seen this before. So I have to make antibodies and other immune cells like memory cells so that I will be able to recognize this when I see this again, which would be if the coronavirus infects you, the spike proteins on the outside will be the first thing that your immune cells see, and they recognize it by the spike protein on the outside. We have heard a lot too about herd immunity. And what is herd immunity? When someone is vaccinated, we know they're protected against a disease. But many people have underlying health conditions that weaken their immune systems, like cancer or HIV, or they might be immunely compromised in some way. And these people can be still protected if they live among vaccinated persons. When most people in a community are vaccinated, the pathogen has a hard time circulating because most people it encounters are immune, and the more others are vaccinated, the less likely people who are unable to be or are unable to take the vaccine are at risk of being exposed to the pathogen. And this is called herd immunity. Okay, so that just takes you through a little bit of information um, on one topic that is considered difficult in biology. But the book, it gives you lots of information that can help you, and I have taken you through some of the information. So I hope I have brought something to pique your interest today and something that you will be able to use. So thanks for your time, and I am available to take questions.
Shall we run the video? See if
Yes.
it works. Let's try. It didn't work so well last time, so I hope it works this time.
Today we are
Today
going to
we
do
are
an
going to do an
experiment
experiment
to look at
to look at
osmosis.
osmosis.
Osmosis refers
Osmosis
to the
refers to
movement
the
of water
movement of water
molecules
molecules
through a
through a
membrane
membrane
that
that
separates
separates
solutions of
solutions of
different
different
concentrations.
concentrations.
Here we
Here
have
we have
a piece of
a piece of
cabbage,
cabbage,
a carrot, and a
carrot, and a
cucumber,
cucumber,
and we have
and we have
grated
grated
the
the
cabbage, the same
cabbage,
amount
the
of
same amount of
cabbage, the
cabbage,
same
the
amount
same
of
amount of
carrot, and
carrot,
the same
and the
amount
same
of
amount of
cucumber.
cucumber.
Now,
Now,
we all
we
love
all love
delicious
delicious
coleslaw
coleslaw
and
and
pickled cucumber,
pickled cucumber,
and we know
and we
that
know that coleslaw is
Coleslaw
made
is
with
made with
grated cabbage
grated
and
cabbage and
carrot, to which sugar has been added, and with pickled cucumber. Grated cucumber has salt added to it along with different types of seasonings. Now, each of these dishes contains the same amount of grated cabbage, carrot, and cucumber, and to the cabbage and the carrot, an equal amount of sugar was added, and it was allowed to stay for 10 minutes, and an equal amount of salt was added to the grated cucumber, and it was allowed to stay for 10 minutes as well. If you don't have any of these vegetables, you can always substitute a marrow or pumpkin for the vegetables. Now, as controls for the cabbage, carrot, and cucumber, no salt was added or sugar was added. And let's now just have a look at each of the dishes where the solute, which was sugar or salt, was added. So for the cabbage, we can see here that liquid has accumulated in the dish. Let me just move it away. You can see that liquid is moving there. Liquid has accumulated in the dish. For the carrot, we can see it there. Liquid has accumulated in the dish. And for the cucumber, we can see all that liquid has accumulated in the dish. Now for the controls, we can see that those are still just moist, but no liquid has accumulated because we did not add any salt or sugar, which we see here in these 2 tablespoons. Now, what has happened? We know that when we grate the cabbage and the carrot and the cucumber, that we damage the cells, and by adding the solute on the outside, which is the sugar or the salt, these cause water to move from inside of the cells, which will be inside of the cell vacuole. So the water travels through the tonoplast, which is the membrane surrounding the plant cell vacuole, and it also, the water moves from the inside of the cell through the tonoplast and also through the plasma membrane, and then it comes outside of the cell and accumulates. Depending on the amount of solute that has been added, the cell might also lose water from its cytoplasm and wilt. And you know this because the crunchy carrot and cabbage that you would have at the beginning, by the time it is in the coleslaw, it is soft and pliable when it is wilted. The same with pickle, the cucumber becomes soft and wilted.
Can you turn your microphone back on, Dr. Durant? Yeah, fantastic.
Yes, okay.
Do you want to take that question?
Yes, so I see we have a question here that says, "Hi, does this book cover all topics in sufficient detail to adequately prepare students for the exam? And the answer is yes. It says, while the syllabus has recommended text, any students cannot afford all or have access. Exams have been known to pose questions that may not have been examined to that extent in the textbook. What can teachers remedy to do this? To this, sorry. So yes, the book covers all of the topics in sufficient detail. The topics in the unit 1 and the unit 2 are included, and it is a recommended text by CXC. Those students who cannot afford to buy a whole textbook, Macmillan gives the opportunity where students can rent a textbook or rent access to the textbook for 3 months, 6 months, 12 months, and 24 months at a very small cost. And you can go to Macmillan Caribbean eBooks to see what the cost of that would be. And here, where we are asking that if the exams pose questions that may not have been examined to an extent in the textbook. Well, certainly when questions are set, the opportunity is taken to test students' ability to apply information that they would have learned in a book or in school to some new variable. So the worksheet, which is being provided by Macmillan, which I designed and is going to be given out to all the participants, that has questions on it which are not directly related to something specifically taken from the textbook, but it has questions that extrapolate from information that students would need to know, and students can use these to practice. Also, the syllabus, the CXC syllabus, which can be downloaded from the CXC website free, has at the end of it past papers and past questions with the mark scheme included, which has in the answers for the questions. And students can use this, and teachers can use it as well to find out, or to see how the questions are designed and what marks are allocated to specific types of answers.
Thank you. That's a very fulsome answer. And has anybody got any more questions or any comments on what Dr. Durant has said? We've got a few more minutes.